Hi everyone, welcome back to Pad Demystified. I'm Dr. Shubhika Sachdeva and today we are going to briefly talk about intracellular accumulations. Now, this is going to be part 1 of the 3 mini series on intracellular accumulations, and today we are going to mainly concentrate on accumulations of lipid. So, what are going to be our objectives in today's small discussion? I'll give you a brief introduction about intracellular accumulations. Then we are going to see the mechanisms of intracellular accumulations, and then we are going to talk in detail about accumulation of lipids. In this, we are going to see this topic under 3 main headings: accumulation of neutral fat, that is your fatty acid and triglycerides, then accumulation of cholesterol and cholesterol esters, and finally, we are going to very briefly talk about accumulation of phospholipids. So now, what are intracellular accumulations? See, the word itself is telling you intracellular accumulation. That means accumulations of abnormal substances within the cell. Now, these accumulations can be within the cytoplasm of the cell or certain organelles in the cells, especially the endoplasmic reticulum or the lysosomes, or they can also occur within the nucleus of the cells. Now, why it is happening? See, these are happening as a result of an intrinsic cellular dysfunction or certain kind of metabolic derangements within the cells, because of which there is accumulations of abnormal products of metabolism within the cell. Now, these accumulations can be of transient and reversible nature, or they can even be permanent. Why? It basically depends upon the amount and the nature of the accumulation. If the substance that is getting accumulated in a lesser amount and it is less toxic, then this manifestation can also be reversible. But if it is getting accumulated in a higher amount and the substance that is getting accumulated is very toxic, then this type of accumulation is usually permanent within the cell and can cause injuries in the cell. So, having understood that what are intracellular accumulations, let us now further go on to the mechanisms of intracellular accumulations. Now, we have 4 main mechanisms which lead to abnormal accumulations of substances within the cells. Okay, so the first mechanism is inadequate removal of normal substances or normal constituents of cell. Now, what are these normal constituents? The normal constituents of cells which can get accumulated includes lipid, proteins, and carbohydrates. Now, why is this happening? See, these substances, they undergo certain cellular metabolism and they produce certain byproducts of that cellular metabolism. Now, these substances are required to get out of the cell and go to their target organs where they can function. For example, lipids will produce fatty acids, your carbohydrates will produce glucose or glycogen, and all these are required to get out of the cells and then go to their target organs to function. Now, if I tell you that there is some defect in the packaging and in the transport of these normal constituents, will these get out of the cell and go to their target organs? Obviously, they will not be able to get out. And when they are not able to get out, that is, there is inadequate removal of these normal substances from the cells, they tend to get accumulated within the cell. So, this is the very first mechanism for accumulation of normal substances. Now, the second mechanism is again because of defect in folding and packaging and transport of certain substances out of the cell. But here, these substances are not normal. These are abnormal substances. Now, why are these abnormals? These substances may be because of certain kind of genetic mutation or certain kind of acquired mutation which might lead to misfolding of proteins, and therefore these proteins become abnormal, and when they are abnormal they will not be getting out of the cell and get accumulated within the cell. And this kind of accumulation mainly occurs within the endoplasmic reticulum of the cell. So the second mechanism is defect in folding, packaging and transport of abnormal substances within the cell. And the classical example for this kind of accumulation is alpha 1 antitrypsin deficiency. So now the third mechanism is accumulation of metabolites because of enzyme deficiencies. Now see what is happening. Normally there is a complex substrate within the cell and there is an enzyme which is helping that complex substrate convert into a soluble product. Now if I tell you that we don't have enzyme at all, there is lack of enzymes in a certain cell. Now if this enzyme is not there, the complex substrate will not be able to form a soluble product. So therefore it is not getting degraded into a soluble product. And because it is not getting degraded, the metabolites of these insoluble complex substrate are getting retained within the cell. There is increased amount of substrate in the cell and it is getting accumulated within the cell. And the classical example of such kind of accumulation is lysosomal storage diseases and inborn errors of metabolism. Okay. And the last mechanism is accumulation of pigments. Now why this is happening? See, pigments are indigestible materials, okay? So either there is no enzymatic mechanism to degrade these pigments into soluble products, or these pigments should get out of the body and go to other sites for function, and there is no transport mechanism for this. So either of the two, if it is happening, these pigments are getting accumulated within the cell and therefore leading to intracellular accumulations. Now these pigments can be either endogenous pigments, that means which are produced within the cell, or these can be also exogenous pigments, that is coming from outside. So, having understood the mechanisms of intracellular accumulation, let us now talk in detail about accumulation of lipids. So, lipids can get accumulated in our body in one of the three forms, either in form of triglycerides or neutral fat, which we classically called as fatty change or steatosis, then in form of cholesterol or cholesterol esters, and finally in form of phospholipids as well. So we are going to first read about steatosis or fatty change, but before I dive into it, let us first understand what normally is happening. So we have certain amount of free fatty acids in our body. Now where are we getting these free fatty acids from? These free fatty acids we are getting from diet in form of chylomicrons, which contain certain amount of triglycerides or phospholipids. And secondly, we are getting these free fatty acids from breakdown of adipose tissue. Once we have these free fatty acids, these go into the liver and form fatty acids. Now this intracellular production of fatty acids, in addition from coming from diet and adipose tissue, some amount of it is also getting converted directly from acetyl-CoA into intracellular fatty acids. Now these fatty acids, majority of them, say 90% of them, are esterified by the action of alpha glycerophosphate into triglycerides. Okay, but say some 5 to 10% of these fatty acids are also oxidized to cholesterol esters, phospholipid and ketone bodies, and these are utilized by our body for our energy needs. Okay, now once the triglycerides are formed, these triglycerides are now converted to lipoprotein by the action of some lipid acceptor protein. Now the example of this lipid acceptor protein is apoprotein. Okay, so triglycerides are then converted to lipoproteins, and these lipoproteins are then finally released from the liver into the plasma, forming plasma lipoproteins in form of LDL, VLDL and HDL. So this is how the normal fat metabolism in our body is going on in brief. So, once we know the basic fat metabolism in our body, it becomes easier for us to understand that defect or derangement in any one of the 6 steps in the fat metabolism can lead to accumulation of these triglycerides inside the liver. That is, intracellular accumulation of triglycerides into the liver can occur in case of any derangement or defect in any one of the steps in fat metabolism. So the first is increased entry of lipids into the liver. Now obviously when I say that there is increased entry of lipids into the liver, I mean that there is increased in free fatty acids, and this can happen in case of obesity or diabetes mellitus, increased alcohol consumption, and in general increased consumption of fat rich foods. The second mechanism is increased synthesis of fatty acids by hepatocytes. Now, this increased synthesis of fatty acids by hepatocytes is classically seen in case of diabetes mellitus. Now, what is happening in diabetes? We know that diabetes mellitus is a state of increased glucose in our body. And when we have increased glucose in our body, there is increase in amount of glycolysis. What does that mean? That means that the increased glucose is getting converted to pyruvate, and this pyruvate is getting converted to acetyl-CoA. So therefore, when there is a condition with increased glucose in our body, it ultimately leads to increased amount of acetyl-CoA in our body. And once this acetyl-CoA is increased, this acetyl-CoA gets rapidly converted to fatty acids inside the liver. So this is the second mechanism. The third mechanism is increase in alpha glycerophosphate activity. Once this alpha glycerophosphate activity is increased, that means there is increased conversion of fatty acids into triglycerides, and this mechanism is again classically seen in alcohol. The fourth mechanism is there is decreased oxidation of fatty acids into ketone bodies and phospholipids. And when there is decreased oxidation of fatty acids into ketone bodies, that means the fatty acids are again getting converted mainly into triglycerides. And this is again seen mainly in case of alcohol consumption, certain amount of liver toxins and hypoxia. The fifth mechanism is that there is decreased synthesis of apoprotein. When there is less amount of apoprotein, again the triglycerides are not getting converted to lipoproteins, and then there is accumulation of triglycerides again. This is classically seen in cases of protein energy malnutrition. And the last mechanism is there is block in excretion of lipoproteins out of the liver. So there is decreased amount of plasma lipoproteins because this excretion is blocked, and when this is blocked there is accumulation of lipoproteins and in turn triglycerides into the liver, and this again is classically seen in alcohol. So the important point here to remember is that alcohol plays a multifactorial role in formation of fatty liver or steatosis. So fatty change or steatosis is also called as fat metamorphosis. Like I have told you before, it is a result of a reversible injury or abnormality in the fat metabolism, which is characterized by intracellular accumulation of triglycerides or neutral fat within the parenchymal cells. And the most common organ affected by fatty change is liver. Why? Because liver is a major organ for fat metabolism. But having said that, there are other non-fatty tissues such as heart, skeletal muscles and kidney which can also get affected by steatosis. So grossly, the liver, if affected by steatosis, is enlarged. It has a very thin but a very tense and glistening capsule. As you can see, the margins are rounded, and once we give a cut through this liver, the cut surface is very yellow or yellowish pale, and it is very greasy to touch because of fat. So microscopically these appear as vacuoles of lipid content within the cell. So why empty vacuoles? You would see there are empty vacuoles over here. But fat was yellow in color, remember, in gross. So what happens is fat in general dissolves with when we treat it with organic solvents. So under the microscope we do not see this yellowish appearance. Instead we see large empty vacuoles with a periphery placed nucleus, and this is what contains lipid into it. So initially these lipid vacuoles are very smaller in size. Okay, you see here, can you appreciate these small lipid vacuoles? This is what we call as a microvesicular steatosis. And when this fatty change progresses and there is more accumulation of triglycerides into the cell, these small vacuoles will coalesce with each other and form a large lipid vacuole with a periphery placed nucleus, and this is now what you call as macrovesicular steatosis. Now a certain affected organ can show you both micro and macrovesicular change within the same section. So here if you see the microscopic picture, this is the large lipid vacuole with a peripherally placed nucleus. Again you see a large lipid vacuole with a peripherally placed nucleus. Let me find some microvesicles for you here. See here, this is a small microvesicular, this is a small microvesicular, and again this is a large macrovesicular steatosis. The subset here shows a special stain which help us to identify that this is indeed fat only, and this stain is called as oil red O stain. Okay. So now, what is the etiology of steatosis or fatty change? So first, any condition with excess amount of fat can lead to steatosis. Why? Because it is beyond the capacity of the liver to metabolize such big amount of fat, and it can be seen in conditions such as obesity, diabetes mellitus, congenital hyperlipidemia. The second is any condition leading to liver cell damage. If there is injury to hepatocytes of the liver, the fat cannot be metabolized properly, and the most common example of that is alcoholic liver disease. The other causes for liver cell damage can include starvation, protein malnutrition, certain amounts of hepatotoxins, and certain drugs producing liver injury. If you recall, we have studied all this in a very systematic manner while studying the mechanism and the pathogenesis of fatty liver. So that was all about accumulation of triglycerides or neutral fat. The second topic is accumulation of cholesterol or cholesterol esters. Now these accumulates mostly in phagocytic cells due to lipid overload, and microscopically these also appear as intracellular vacuoles. What is the etiology for accumulation of cholesterol and cholesterol esters? The very first condition in which there is accumulation of cholesterol is atherosclerosis. See, what is happening in atherosclerosis? There is accumulation of lipid-laden macrophages within the intimal layer of the blood vessel. You see, concentrate on this particular cell over here. It's a large cell with a clear cytoplasm, and in between this cell there are small lipid laden vacuoles, and this particular cell is what we call as a foam cell, and these foam cells are very characteristic of a fully developed erythematous plaque. The second condition in which cholesterol can get accumulated are xanthomas. Now, xanthomas are characteristic of an hereditary hyperlipidemic state, and here also you can appreciate big macrophages, and in between those macrophages there are accumulation of cholesterol laden lipid vacuoles. The third condition in which it is happening is cholesterosis. Now, cholesterosis is predominantly seen in the lamina propria of the gallbladder. See, this is a section of a gallbladder. This is a gallbladder lining, and beneath that there is the lamina propria. And in the lamina propria, you can appreciate there is a big cluster of macrophages over here. And these macrophages are laden with cholesterol and cholesterol esters. And the last condition is Niemann-Pick disease. Now, this is a type of autosomal recessive lysosomal storage disease in which again there is accumulation of macrophages within which there are cholesterol and cholesterol esters. Why this is happening? In this, there is an inherited impairment of cholesterol transport out of the lysosome, and these children developed central nervous symptoms and very progressive, and they deteriorate very progressively. So now lastly we have accumulation of phospholipids. Okay, phospholipids are a major component of a plasma membrane or the cell membrane. So when you have a cell injury which is damaging the cell membrane, these phospholipids are abundantly released, and the accumulation of these phospholipids are called as myelin figures, and these myelin figures accumulate within the cytoplasm of the cells, and eventually they are degraded into fatty acids, or they are phagocytosed when finally the cell dies. So now we have read about fatty change, accumulation of cholesterol and cholesterol esters, and finally accumulation of phospholipids. Microscopically, though the etiology for each and every one was different, microscopically they appeared as vacuoles within the cell cytoplasm. Now, it is very common for us to confuse these lipid-filled vacuoles with an other reversible change, that is the hydrophic change. So how do we differentiate that this is not an hydrophic change, this is a fat? So we have certain special stains which will stain especially the fat in a fresh unfixed tissue. And the very first stain for that we have is a Sudan black. You see, this special stain is staining the fat into black color. The second stain that we have is oil red O stain. You see, it is staining fat into bright red color vacuoles. So this is how we demonstrate fat in any fresh or fixed tissue. And the last stain that we have is an osmic acid, which is a fixative as well as a stain which can demonstrate fat in certain tissues. So that was all about intracellular accumulations in general, accumulations of lipids in detail. In the subsequent series in part 2, we are going to read about accumulations of proteins and carbohydrates, and finally accumulation of pigments inside the cell. So that's it for today. I hope that you enjoyed this short tutorial on intracellular accumulations and accumulations of lipid. Stay tuned for many more exciting videos, and don't forget to like, share and subscribe to Pat Demystified. This is me signing out, and I'll see you in the next video soon. Bye.